Hey students, welcome back for part 2 of our lecture regarding topics 2.3 and 2.4, looking at the nervous system and neural transmission. We're going to focus on these next two learning targets here, describing the parts of the neuron and identifying the basic process of transmission of signals between neurons. So I have a little neuron quiz here for you. I want you to try to match these. So go ahead, pause the video now. So here are the answers. Hopefully you got those right, and if you didn't, it's okay. You should know these by the end of this lecture. So let's talk about neurons, okay? Neuron cells, what are nerves, and what are glial cells? So we're going to talk about neurons, and you'll also need to make sure you understand what a glial cell is as well, as they're very important in the anatomy of a neuron. So the body's information system is the nervous system, right, which we talked about in our last part of this lecture. We talked about the electrochemical communication system and how our body uses the nervous system to communicate things about movement, thoughts, internal organ functions, sensations, perceptions, all these things. And they're made up of these basic cells called neurons, okay? And while they're very basic, they're also very complex, and they have three functions, okay? To receive information, to carry information, and to pass information. And then you have nerves, right? Nerves are made up of these neurons, right? But they are much larger bundles, okay? So they're not a singular neuron, but a large bundle of neuron axons outside the brain, the spinal column. The nerves that are located inside the brain would be cranial nerves, okay? But also you have another type of cell listed here, and that would be the glial cell, known as the worker bees, okay? And the reason we call them the worker bees is because they provide nutrients, they insulate the myelin, they guide neural connections, they nourish the neuron, and they protect the nerve cells. So you can see here, in this picture, they are the green cells here, whereas this purple cell here would be the neuron, and this is the axon, okay? The long, thin part, and then you have these glial cells supporting the myelin, right, which insulates and protects it. So they're known as the worker bees, and we have about 10 to 1 glial cells to our neurons. So we have a lot more glial cells in our body than we do neurons. Now, next I want to look at the neural communication. So we're going to talk about the anatomy of the neuron and all the different parts. So make sure you have your book here with you. If you don't catch everything I'm saying, the book right here, it's on page 82. So if you miss anything, you can look there or follow along. But the first part we have, and we're going to talk about, is the dendrites. So these are these little tree-like branches out here, and they listen and pass on information to the cell body. And they're really quite incredible, as a neuron can have hundreds or even thousands of these little dendrite spines, and they can also connect to various different neurons. So they're pretty remarkable. Generally, the more developed a neuron is, the more of those dendrite spines it'll have. Next, you have the cell body, okay? The cell body here, and this is where the dendrites are going to send that message, that information to. And this is the large portion of the neuron that contains the nucleus here in the middle, right? The nucleus makes the decision to fire or not to fire. So the cell body is the life source of the neuron. Next, you have the axon, okay? The axon is the longest part of the neuron and is the thin fiber that speaks, okay? And it sends a neural impulse of communication down this neuron, neuron. It's the longest, thinnest part. And then you have the terminal branches down here at the end, okay? This is the end point of the neuron, and this is where the synapse is going to occur, where neurotransmitters are going to be passed on to the next neuron through these little bulbs or these buds, okay, at the end. And then you have the myelin sheath, okay? The myelin sheath is the fat, tissue-y based thing that kind of protects the axon and helps keep it insulated and keeping that electrical pulse traveling directly down to the terminal branches efficiently and effectively, okay? So if you think about like a wire, right, an electrical wire cord, okay, generally there's that coating around it, right? There's insulation around it, and that rubber coating on the outside is meant to keep that electrical charge going through. If it gets out, if that plastic starts to come off or that rubber starts to come off the cord, then it loses that insulation and the cord doesn't work as efficiently, right? Same thing with our neurons. And the last structure I want to talk about here for the neuron would be the nodes of Ranvier. It's just named after the guy who discovered it, but it's these little spaces in between, or these gaps in between the myelin sheath, and the function of it is to keep that charge really going through the axon. Without them, the charge could possibly lose its intensity before it reaches the cell that it's trying to communicate with down the line. Okay. Now the myelin sheath protects that neuron, right? And it's interesting because there are two different types of myelin that you'll see in the peripheral nervous system versus what you see in the central nervous system. So kind of as I spoke about how in the central nervous system, generally if you damage those axons, they don't really regenerate, right? Whereas in the peripheral nervous system, if you damage neurons in the peripheral system, they do regenerate. That's a major difference between these types of myelins that are protecting those axons. Now, in the peripheral nervous system, you have what's known as the Schwann cells. Okay, so the Schwann cells create this sheath in the peripheral nervous system, and they only cover one segment of that axon. As you can see, you've got a Schwann cell here, you've got a Schwann cell here, you've got a Schwann cell here, up amongst each segment of that neuron. Okay, and again, these are made up of glial cells as well, as those glial cells help nourish that myelin sheath. Okay, and then over here you have the central nervous system, and the central nervous system is made up of, well, the central nervous system's myelin is made up of oligodendrites. Oligodendrites create the myelin sheath in the central nervous system, and they can cover up to from 3 to 50 neurons. So while the Schwann cells are just going to cover one little segment of that axon, over here it's going to cover the entire axon as well as possibly 50 other axons. Okay, if this is damaged, then it's going to make for a very difficult time recovering from injuries and healing. These are the different types of cells that help generate and create these myelins to protect our axons. Now I'm going to post this video for you guys on the Google Doc, but it is a video about multiple sclerosis and how the myelin sheath, while it acts as that function to insulate our axons and keep that neural transmission going smoothly, some people have something called multiple sclerosis where that axon is not able to communicate very well with other cells because of the myelin sheath's degeneration, and you have loss of muscle control. Now that we've looked at the anatomy of the axon, or the neuron, let's talk about the neural impulse and how this neuron actually communicates in our bodies. You all have been in science classes before, and you learned that cell bodies contain ions, right? Well, the same thing goes for neurons, right? You've got positive ions and you've got negative ions. You've got positive charges and negative charges. So therefore, when a neuron is in its resting state, the neuron has a negative charge. And that negative charge at resting state is about 70 millivolts. Okay, we call this resting potential. Now, the reason we know about neural impulses is due to Alan Hodgkin and Andrew Huxley's research on squids and squids' axons. The reason they used squid is because squids have an axon that is about 100 times bigger than humans, so it's just easier to experiment on. But the squid's axon functions almost identical to that of a human, so it really has helped unravel the mystery behind the neuron. Now, how does this neuron actually fire when we look at a threshold? What is a threshold? A threshold is the minimum level of stimulation required to trigger that neural impulse. So for that neuron to leave its resting potential and have an actual action potential, it's got to break that threshold, okay? The minimum stimulation. So the minimum stimulation for an action potential to occur would be about 30 millivolts. And then after it ends through that action potential, it goes through the repolarization phase and then back into the refractory period where it recharges so that it can fire again. So this is a good chart and an example for you to kind of see what it looks like. And this is how quickly it would go, right? I mean, that's extremely fast. That's about how fast your neurons can fire, right? Every time that's blinking, it's fired again. So here we have the electrical component of what's actually going inside, what's going on inside of this neuron. And I have three terms up here: polarized, depolarized, and refractory period. So the polarized resting state, right, is when the neuron is in a stable negative inactive phase. Okay, so it's negatively charged on the inside, positively charged on the outside, right? When the neuron is then stimulated, channels of that cell membrane are going to open up, and for a very brief moment, it's going to allow positively charged ions to rush in, and then the negatively charged potassium ions will go out, and then more positively charged sodium ions will come in, and then more potassium negatively charged ions will go out, okay, as you can see here. So as this sodium comes in, the potassium goes out, and again, as more sodium comes in, the potassium comes out, etc., etc. Now here, our polarized membrane is at its resting potential. So we have all negative charges within inside this axon membrane, okay? And if we look over here at this chart, we see our resting state at negative 70 millivolts. It has that threshold here at about negative 55 millivolts. Once it reaches that, okay, once it reaches this, it fires. Once it reaches that threshold, it's able to fire, and it depolarizes. So this right here is the process of depolarization. As positive ions come in, negative ions go out. Okay, so depolarization is this cause of action potential. It's when the positive sodium ions move in, and this creates an electrical friction, and the neuron then becomes positively charged. It's like a spike of voltage, okay, and it's generally very fast. And then you have hyperpolarization or repolarization, as you see on the decline here, right? And this is when the negative ions kind of enter back into the neuron, making it less prone to firing the next action potential. And then lastly, it goes in here to the refractory period, okay, and the refractory period is the recharging period when the neuron cannot fire for a very short period of time, only about 1 to 2 milliseconds before that neuron is recharged and ready to fire again. So if you think about like a camera, when you take a picture, right, with your camera and it flashes, right, it needs a second to recharge, okay? That's the refractory period. Right, so within the neural impulse, you have this principle called the all or none principle. Make sure you know this. It is the principle that if a neuron fires, it will always fire at the exact same intensity. Okay, so all action potentials are the same strength. A neuron does not fire at 30%, does not fire at 45%, does not fire at 90%, fires at 100% every single time. And so we have this thing called the toilet analogy, okay? And the toilet analogy just tells us that, like a neuron, a toilet has an action potential, okay? When you flush that toilet, an impulse is sent down that sewer pipe, all right? And then you have the refractory period, right? There's a short delay after flushing that toilet where you cannot flush again. And then you have the resting potential when that toilet is now charged and ready to go to be used again, okay? This is the all or none principle. It either flushes everything down that toilet or it doesn't flush. It can't flush at 30%, okay? Either it's flushing everything down or it is not flushing at all. It either works or it doesn't work. Same thing goes with our neurons. They either work or they don't work. All right, so go ahead and review the process of a neuron, the sequence of neural transmission. I want you to try to put these in the correct order. So pause me for a second, and then I'll show you the answer here briefly. All right, and there are your answers, okay? So the axon is in its resting state. The interior is negative. Goes to sodium channels opening up, and then those sodium ions flow into the axon, making the interior positive. Next, you have potassium channels opening. Potassium ions flow out of the axon, making the interior negative. Sodium potassium pumps then restore that proper ions ratios back to normal. You go back to your resting state. This is the sequence of the neural transmission. Next, we're going to talk about communication between the neurons. So let's get into that. So when we talk about the synapse, okay, the synapse is the junction where information is transmitted from one neuron to the next neuron. Okay, and within this synapse, you have something called the synaptic cleft. Okay, they're different. The synapse and synaptic cleft are different. Sometimes students confuse those on the tests. But this is the tiny microscopic gap between the neuron and the cell membrane of another neuron. Signals have to cross this little cleft, this little gap, to permit those neurons down further to other receiving neurons. So in this situation, the neuron that sends the signal across the gap is the presynaptic gap. So if you look here, you've got your axon, you've got your terminal bulb here, and then here is your synaptic cleft, okay? This little tiny gap between these two, and here you have your presynaptic gap, there's your presynaptic membrane and your postsynaptic membrane, right? The one that would be receiving. So presynaptic is the small surface in the synaptic terminal that faces the synaptic gap and releases the neurotransmitters into that gap, and then the postsynaptic gap is going to be, the postsynaptic membrane is going to be the one that receives it, right? Receives the neurotransmitters through those dendrites and then passes that message on down to the cell body and then to the axon and then same thing, okay? Over and over and over, right? Also, you have the synaptic vessels as well. Make sure you know those. The synaptic vessels here, there's little red bubbles inside of here, okay? These are the areas that hold the neurotransmitters until they're released for that presynaptic membrane to send them on to the next neuron. So if you think about learning and anything that we learn, like learning how to write, learning how to type, learning how to brush your teeth, learning how to walk, right? This synaptic transmission has to happen in order for you to be able to do those things. And if you've ever thought about, hey, the first time I learned how to write my name on paper, right? It was hard, it was challenging, it was difficult. Now you do it, I mean, without even thinking, right? It's very easy for you. But the first time you did that, that took a lot of thought and a lot of concentration to be able to do that. The more you do something, right, the more this synaptic transmission happens. It's much smoother, right? It's because you have more receiving dendrites on the other side. And this is a developed pathway. It's just like if you think about traveling a hiking trail for the very first time, the very first person that goes on that hiking trail, it's difficult to get through it, right? Because they're the ones who are establishing it for the very first time. But the more it's traveled, the easier it becomes. The same thing goes with our learning. Okay, the more that neuron fires, the more that neural transmission is occurring, the easier it is for your body to communicate that message, for you to write your name on that paper. So now that we understand the synaptic gap, we're going to talk about these chemical messengers. Okay, and these chemical messengers are known as neurotransmitters. So neurotransmitters are the chemical messengers that are passed down by a neuron, and they are released in that synaptic cleft from the presynaptic gap, or from the presynaptic membrane to the postsynaptic membrane. Okay, and these are some of your most common ones that you'll see in psychology: dopamine, serotonin, norepinephrine, acetylcholine, GABA, and endorphins. Now make sure you know these because they will be on your test, and you have a table in your book so you can study them there. Make sure you memorize all of them, all right? Dopamine is going to be related to movement and thought processes. Serotonin is going to be our messenger for emotional state and sleep. Norepinephrine is going to be our messenger for physical arousal, learning, and memory. Acetylcholine is going to be our messenger for muscle contraction, learning, and memory. GABA stands for gamma aminobutyric acid. It is a relaxation messenger, okay? And it's also an inhibitory neurotransmitter. And then endorphins. They're going to be most commonly known as runner's high. Ever heard of runner's high? Okay, they reduce pain and they give us a sense of pleasure as well. And whenever we have something that we've accomplished, right, it boosts our mood. So a test tip would be to know that acetylcholine is actually generally linked to Alzheimer's disease, if you've ever heard of that, okay, regarding memory. And then dopamine is linked to one disorder and also a disease. So too little dopamine is linked to Parkinson's disease, and then too much dopamine is linked to schizophrenia. So they have something called the dopamine hypothesis, which we'll talk about more so when we get into our abnormal psych unit. We'll talk about it more when we get into abnormal psych. Now, the last process we're going to talk about in this synapse, okay, is reuptake. So once the neurotransmitters bind to a receptor site, key and lock, right? The key is going to be the neurotransmitter. The lock is going to be right here. The receptor site has to fit hand in glove, right, for that message to be able to pass on and be communicated. Some molecules will be left in that gap. So this is called reuptake. It's a process in which our neurotransmitters are basically sponged back up from the synaptic cleft by that presynaptic membrane, okay? And it's our body's way of recycling these neurotransmitters that doesn't need to use, okay? So it'll use them for later. It doesn't want to overload the receiving neuron. It wants it to be the correct amount of messages. So our body has this perfect combination, and it knows when too many neurotransmitters are being received, and so therefore it reabsorbs those. Now, you do have some drugs that actually block reuptake. One of those drugs would be Prozac. I'm sure you've all heard of what Prozac is, but what it does is it actually blocks the reuptake of the neurotransmitter serotonin so that it can keep serotonin in that synapse for a longer period of time and help that individual elevate their mood, okay? So it will help with things like depression. Now, of course, there are side effects to all of these different drugs, and it can affect other neurotransmitters as well. But Prozac is something that is generally pretty commonly prescribed for mood boosting for those who deal with depression. And then lastly, we're going to end today with an AP FRQ exam practice. I'll leave you time to go ahead and pause the video here and read this. Post your response to this video on PowerSchool. Make sure you bold and underline and space out your answer for each segment of the answer. Part A, Part B, and Part C. All right, that's it for today, guys. I'll see you next time.